Okay, everybody ready? Perfect. So I wanted to make sure that we covered steroid messengers well enough from yesterday. So let's just revisit a last couple of the slides that we did yesterday. Remember that messengers are, again, these chemical signals that are just going to go back and forth between different places and initiate oftentimes these signaling cascades. If there's steroids, your receptor is going to be inside the cell, so steroids are able to go through that lipid bilayer typically, and so the receptor is inside the cell as opposed to water-soluble molecules where it's on the outside of the cell. And then you end up with a second messenger type situation. So oftentimes, the steroid hormone comes through the lipid bilayer, interacts with its receptor. It may or may not have what's called a secondary messenger. And remember, we saw that those secondary messengers are often like cyclic AMP, cyclic GMP. And then eventually, you get an interaction with DNA, and it changes something about the protein. It changes something that that cell is going to do. So that's how the steroid messengers work. Remember, these encompass the whole category of sex hormones, which all are synthesized from cholesterol. And you start out with cholesterol, and it gets synthesized to progesterone, and then progesterone goes on into these three different molecules here. And those are responsible for the development of those secondary sexual characteristics. So these, again, are very, very important. We talked a little bit yesterday about why progesterone is so important in development and in labor. Remember, progesterone is that molecule that you really need at a sufficient concentration to maintain a pregnancy. Oftentimes, we see drops in progesterone, especially in other species, not in humans, actually are what's causing the initiation of labor. And so there's some thought that progesterone is part of what's causing preterm labor. It's something that in the developing world, we still rank near the bottom in preterm labor. This is something that really plagues developing nations and developed nations as well. So progesterone in humans is not shown to decrease, but what ends up happening is you get this inflammatory response, and you have prostaglandin 2 that gets upregulated, and they think that that has a lot to do with the initiation of labor. So here's a prostaglandin type reaction, and so this is a specific type of membrane receptor. It's the FMLP receptor family, and you can see these transmembrane receptors. So whenever something goes through the membrane, that's a transmembrane receptor. And then you can see some of the signaling cascades here, and here's prostaglandin, and you go through the biological effects. So remember, we just talked about steroid messengers, but there's also chemical messengers. Chemical messengers, steroid messengers are kind of just a small class of these chemical messengers, but chemical messengers are classified according to how they work. So they can either activate enzymes, affect the synthesis of enzymes, so those are both enzymatic type effects. They can affect the permeability of the membrane, so how big are the molecules that can get in and out of that cell. And then they can act either directly or through a secondary messenger. And again, these are mostly hormones. We've got our hormone here and our receptor. We've got a secondary message. It goes over to this adenylate cyclase, which produces another message. And so this is what we call a signal amplification cascade. Yep, we're going to talk a lot about that tomorrow. So then keep in mind that when we talk about drugs, we're talking usually about either antagonists, meaning that they oppose the action of a drug, so they block a receptor and prevent its stimulation, or agonists. And agonists, they compete with a natural messenger for a receptor site, and once there, it stimulates the receptor to do something. There are certain drugs that can increase concentration of a messenger, and again, that is going to inhibit that messenger from actually disassociating from the receptor. And so that drug stays in the system a little bit longer. You also get other drugs that either inhibit or activate specific enzymes inside the cell. So again, a large number of the drugs that we encounter are going to fall into one of these categories. And oftentimes you'll be plagued by the words antagonist and agonist throughout your PharmD careers.
So one thing, one other point, a lot of drugs are straight up hormones. So you'll see things like testosterone, estrogen, progesterone. Estrogen will beat the drugs themselves. So a hormone replacement or treatment for whatever disease. So those will be straight up drugs a lot of times.
Well, and the interesting part about these hormone drugs that we didn't really address here, hormone drugs, these hormones are really, really short half-lives. And hormones get bound up by a lot of proteins that are just floating around in your blood and your serum. So like progesterone has, I think it's a 90-second half-life. I mean, it gets turned over really fast. And it's about 90% bound up by proteins. So that means only 10% of it is bioavailable for the action that you need it for. And so when we prescribe progesterone, we either have a really high dosing frequency, or you give a lot more than what's actually necessary for the action, because you know that not much of it is going to get to the site of action. So this is a problem when we're trying to deliver and when we're trying to use these hormone replacement type drugs. You have to deal with bioavailability, short half-life, and a variety of different things. So that leads us into carbohydrates. And you're going to see these 3 lectures this week are all going to kind of mix together on some level and cross over. But today we're going to deal with carbohydrates. And this is just kind of a funny cartoon that shows us how much people really don't understand about carbohydrates and how they've kind of been vilified in our dietary systems. So let's start with a case study. Jenny Baird is a 34-year-old woman. She comes to the doctor's office with influenza-like symptoms, including a fever, large tender lymph nodes, throat inflammation, a rash on her trunk, frequent headaches, and sores on her mouth. She has a history of asthma, GRED, and frequent bronchitis. What kind of diagnosis would you have? Herpes, good guess. No? Well, it's not the whole story. So let me tell you a little bit more about this. She also was given a blood panel. She tests positive for HIV, and then it was confirmed through a Western blot and RT-PCR, negative for hepatitis, negative for mononucleosis, and negative for Epstein-Barr. So let's talk for just a minute about what those tests look like. So did you guys know that there is an at-home diagnostic for HIV? You can actually go to your pharmacy and get a test for HIV. Is that a good thing or a bad thing? Okay, good. Why good? Accessible, yeah. A lot of people delay testing. They don't want to be tested because they're afraid and they're concerned about stigmas. Is there any part that would be bad? False positives, yeah. So we're going to talk about that. False positives are very bad. How many of you understand what a false positive means completely? How many of you would know if you got a false positive on your test? It'd be really hard to know. Yeah, no, I mean, it would be very hard to know. So the point is, it doesn't violate HIPAA.
Yeah, I mean, if you ask them a question, it doesn't dictate.
No, the point is that you guys are educated. But think about, not all of your patient population is going to be as educated as you are. So if somebody got a false positive, how would they know that it was a false positive? Do they even know what a false positive means? And then...
This is a great question. This actually happened to me in class.
You violated HIPAA? Oh, no, sorry. Oh.
So what ended up happening is I gave this lecture in class, topic and one of the I took an HIV test. I got positive. I know I wasn't positive. That's a false positive. The test says you're positive, you're not positive. False negative is you're positive, the test says you're negative. Right? So all these tests are geared towards minimizing false negatives, right?
Because you want to screen for people. You would rather people think they're positive and act accordingly than think they're negative and act accordingly.
There's a lot of people, there's like actually a pretty high false positive rate on HIV tests.
Well, and we're going to talk about that here in just a few minutes.
I'll just say this is a real
It
life
happens.
scenario. This woman was like, she almost lost her job, or they had to go through a bunch of review process to make sure because you don't want a nurse who's HIV positive in all scenarios, right? You can imagine that being sort of keep drawing blood and stuff. It's not always a great scenario for a nurse. So it was very difficult, and obviously it was very challenging for their marriage and other things, right? So, and then I'll talk about how this works and how this happens.
Yeah, so I just want you to think for a minute. Put yourself, you're in the pharmacy, somebody comes in and says, I got a positive on my HIV test. How do you counsel them? What do you say? As a pharmacist, you might encounter this at some point. If somebody says, I need to buy an HIV test, but how do I take it? What do I do? These are all questions that you may be asked at some point. So we can put the power for testing in the hands of consumers, but at the same time, we need to make sure that there's some education that goes along with it, right? So this is how the HIV test works. Now this is a first generation one. They've gotten increasingly more sophisticated, but this is an ELISA-based test. Did we talk about ELISAs in this class? We did, right? Okay.
You should know what ELISA is. You know the title. You should understand that.
Yeah. Know, what's the acronym ELISA stand for?
How it works.
So here's an ELISA, and we put an HIV protein, which is an antigen, down on the surface of a 96-well plate. So this is just a plastic plate. It's called a microtiter plate. And then we take an antibody specific to the HIV protein. And this is HIV-1 protein. That doesn't really matter for this part, but it's going to interact with the antigen. Then we have to detect it when that action has happened, when that interaction has happened. So then we take an antibody against the first antibody, and we put a tag on the end of it, and we say, okay, this tag is now going to act with a substrate to give a colorimetric change. That's how this ELISA is going to work. So what people see in this first generation test is they put their sample down and they do some washing and they try to figure out, is there a color change?
So just a note, the medicine, the tag, that's actually an enzyme called horseradish HRP. That enzyme is exactly what we talked about last week. It's an enzyme that chews up a substrate.
It's going to cut up this substrate to produce a color.
And then what magically happens is that well turns into color. You can visually see that.
It's very similar, yeah.
Almost exactly the same technology.
So we're showing you an ELISA. Really what this test is a lateral flow device. And that's beyond the scope of this class. But I got to save some secrets for pharmaceutical biotech because we spend like a week on lateral flow devices. But essentially just think of it as a mini ELISA put on a stick, and you're using chromatography in order to separate things. Okay, so
We use the same enzymes, we use the same,
yep,
all the
same
same
substrate.
all the same technologies in the lateral flow test. with ELISA.
So this is what that plate might end up looking like, and then they do a Western blot in order to confirm the presence or absence. And you should note, look at what we're looking at here. Here's glycoprotein 160, glycoprotein 120, glycoprotein 41. These are going to become very important later in today's lecture. So now we are getting a little bit more sophisticated. And you end up having a different combination of antibodies, which gives you increased specificity and increased sensitivity. So here are a couple of the different types of testing kits available. This one, anything that has this asterisk, means that this is kind of a point-of-care test that a consumer could actually go and pick up and do. Or it's something that's going to be done in a doctor's office at that time, like a strep test. And so again, the OraQuick is the one that most people know about in terms of what's being sold in a pharmacy. And that's going to use saliva, whole blood, whatever. But look at all the things that can actually cause a false positive. Now this becomes a huge issue. Particularly, this is a particular problem in Africa where you have a lot of these things that are also occurring. But look about, tuberculosis, herpes simplex 1 and 2. Just having a cold or a flu when you take the test can give you a false positive. Malaria can. Pregnancy, if you're carrying more than one baby, you can actually get a false positive HIV test. So we can do, all sorts of different things. Hepatitis will give you this. So this is an issue. This is a problem. So we've got, going back to our case study, we've got this woman who comes in, flu-like symptoms, rash on her body, and we give her this series of blood tests, and we say, oh, you're HIV positive. So we said that she was negative for Epstein-Barr, which is the cause of mononucleosis. So we actually did two different things. But look at how complicated this scenario is. So there's actually 3 different antibodies showing up here. And it turns out that the interpretation of a mono test is very difficult because you can get a positive here and a positive here, and that means you have had a past infection. You can get a positive here and a positive here. That means you have an acute primary infection. If you get positives in all 3, it means it's indeterminate. And so you have to understand how you are going to interpret this type of test. But in our case study, she was told that she was negative for mono and negative for Epstein-Barr. But now let's take a little bit more history. The rest of the social history is that she's sexually active, has multiple partners. She has a history of crack cocaine use, not within the last month. She smokes marijuana once monthly, is a tobacco user, and has typically 1 to 2 drinks on the weekend. So now knowing this social history, would you think that the HIV test would be a false positive? You would think it would be a false positive? Okay. And why would you think false positive? Yeah, so other drugs in the systems could alter the results. The antibody should be a little bit more specific.
But she also has, just 2 significant risk factors that are also there, right?
Yep.
And
Multiple partners, yeah.
so it's, it's good to ask the question, but it's also good to say, false positive, right?
So the point of dividing that up is don't forget to ask all the questions. Don't forget to ask questions about things other than just medication usage and things other than just this, because maybe you'll find that there's a nutritional deficit or something else, okay? So when you're counseling your patients, make sure that you're asking and getting full history. So that takes us into talking about HIV. Now, HIV is a retrovirus. In fact, it's a specific kind of retrovirus called a lentivirus. And remember, a retrovirus has RNA as its genetic information that it's going to transmit. And then it hijacks your cell's machinery, because remember, viruses are very small, so they don't have all the necessary machinery to replicate by themselves. So it's going to hijack your machinery in order to make your cells replicate it. So here is HIV. Now, HIV is also very tricky because it infects immune cells. And the reason it does that is because of glycosylation. So you've got your envelope, you've got your capsid here that's encapsulating your RNA, You have an integrase, which helps it integrate into your genetic information, a reverse transcriptase, a protease that allows things to break apart. And so you have all these different pieces in a very small package. It's a very, very tiny package. And what we find is that one of the things, HIV seems very scary and retroviruses are very scary, but at the same time, you got to admire the way a virus can replicate. And so people are now starting to look at viruses and saying, can we engineer a virus like this that can infect a cancer and be an oncolytic virus and destroy a cancer specifically? So we could look at viruses as not only things to avoid, but also things to take advantage of. So this is how a retrovirus works. You've got your retrovirus that interacts with a receptor on the cell membrane. It gets endocytosed, uncoated, releases its viral RNA, and then it carries its own reverse transcriptase to make DNA, and then it carries an integrase that allows it to integrate into the DNA of the host. And you get budding and new murine entrants. So this is how HIV specifically enters. You have this docking that occurs here, and it's using a CD4 receptor. And then you have a fusion event similar to the retrovirus here. And from there, I think I'm going to let Ben take over because he's got some pretty cool PyMol stuff to show you about the envelope proteins and the envelope glycoproteins.
Okay, so the important part about today's lecture is obviously about carbohydrates, right? What? Carbohydrates? What do you relate to?
Sugars. I'm just telling you it's sugars.
It's sugars. The sugars. Okay? So your traditional thing about sugars that we talk about is what? We just talk about it in terms of metabolism, right? I eat sugar, I process it, I use it for energy, right? That's generally what we're going to think about in terms of carbohydrates. So I'm going to give you an example here where it's not functioning in that manner. We use carbohydrates actually for a lot of different things, and I think we'll talk a little bit about how they're really important in plants, as structural entities. So I'm going to be honest. I used to hate carbohydrates with a passion because they're just. My wife has an affinity for carbohydrates in many ways. Okay, so do I, but that's a different story. So, but the important part really is I'm going to show you how viruses use carbohydrates to mask how they get in. So GP120, so Amanda just told you that CD-positive T cells are the target for HIV. What ends up happening is HIV goes and infects a CD-positive T cell. T cells kill infections, right? So you don't usually die from HIV necessarily. You die from secondary infections of the immune system. And so what ends up happening here is HIV will come in, and proteins sitting on top of a virion, on top of a virus, are classic markers for your immune system to go, oh, we need to attach to that and clear it. Okay, but on the top, this is GP120. This is a trimer. Okay, but on the top of GP120, and there's a lot of viruses that do this. They start putting on large carbohydrate chains, large sugar chains. Okay, sugars are very non-reactive to the immune system. The immune system doesn't really respond to them. Okay, so on the top of these proteins, we're putting all this forest of sugars, right? And the immune system goes, oh, nothing to see here. Let's move on. And that allows GP120 then to bind to CD4 positive T cells, and then that allows them to infect. So this is called what's called a glycan shield in virology. So if you wanted to know why we have such a hard time with HIV and why the immune system can't do it, can't solve it, this is why. Guess what other diseases or viruses are also use this? What other diseases do we have a hard time with vaccines? Yeah, your flu. So I work at Penn. It was one of my collaborations, and I was there. I work with herpes guys, right? And
down the hall is an influenza guy who actually tests the vaccine for the CDC every year. And they discovered that there was a mutation, and this was actually 2 years ago. There was a mutation in the, so flu has 2 main proteins in it that are on the capsid. And there was a mutation that allowed for an additional glycosylation site, and that was the reason the flu vaccine was not as efficient that year. It's because it added a glycosylation site, and these big sugars here then all of a sudden were able to help evade the immune system. So here's the CD4 binding site, right? And you can see that there's all kinds of spots. This is how it sits outside. This is the viral envelope, and this is how this protein kind of sits out there. And we have this nice structure. And there's a lot of antibodies to these sites right here, right? There's a lot of binding sites that are potentially, because there's a lot of charges on that. And what the virus does is takes and puts carbohydrates all around these things to make it very difficult to see where those protein binding sites are occurring. So antigenic sites are masked by putting all of these carbohydrates, which are in blue here, on those sites. So what I'm going to show you now is the Pymol structure for this. And I'm just going to show you how you could use this in your video. So the model we're going to look at is 1GC1. So I'll do fetch 1GC1. So I did a remove, not polymer. If I go back and I look at the site, oops, 1GC1. What did I mistype here? 1GC1. There we go. So if I look at it, I have an envelope protein, that's G, and then I have a CD4 molecule, that's the receptor, right? And then I also have an antibody. We talked about antibody drugs, heavy and light chain yesterday. So there's actually 4 protein chains, 3 different proteins in this thing. So we're going to go, I just know that. I'm telling you that as that's the receptor on the T cell. CD4 is the protein that GP120 binds to. Excuse me. It's a good question. So well, it depends on what you have, right? So it depends on what if you're talking about, if I'm talking about herpes, right? I know that GD is the fusion protein. That's the protein that's going to bind to the receptor. And I know that the receptors are going to be nectin, right? So you can look that up and just see what if you type in, yeah. Yep. And then what you do is, so then what you do is you can type in HIV, GP120, CD4, PDB, and then this model pops up. And I've done this 500 times. So if you are like lost and you're like, hey, I'm studying XYZ and I need help identifying what the proteins along the way are, I can help you completely do that. That's generally not a big deal. So, but knowing this website here, I mean, the reason I keep doing this is just so you get an idea of how to do it. It's not, so I'm going to label CD4 as chain C, and my envelope protein GP120 is G. So when I come in here, if I type in, I'm going to hide everything. And I'm going to type in select CD4, comma, as chain C. And I'm going to type select GP120, comma, as chain G. So I have both those. I'm going to show them as a surface. Show it as a surface. So hopefully you can start to see, actually I'm going to flip it up just so you can get a better idea. So CD4, I'm going to color that red. Oh, and my GP120 I'm going to turn into gray. So hopefully you can start to see how these two are going to interact. Now, I'm going to show you all the glycosylation sites here. There's a whole boatload of them. I typed them in beforehand because I didn't want to sit and go have someone read off all these sites to me. But if I go select glycosylation, comma, RESI, and all that, and I color them green. Oops, did I? Oh, I didn't put the right, put on the other additional residues too. But you can see all in this molecule. Let me just hide CD40 real quick. You can see on this molecule all the sites that are around that are going to help obscure that molecule. Okay, from the immune system. But it's still, so CD4, that allows it, that binding site is not obscured, right? And so that molecule is then able to bind, and that's how HIV then will come in. So what are we asking you to do here? This would be a great way to present in your video the glycosylation sites just by coloring them a different color and having them stand out if you were doing something like this. If you wanted to do HIV. So it gives you a good idea of how. And you can see all the different residues that are then now colored and are affected by glycosylation. Does anyone have any questions? I'll just, as a quick note, if I hit sequence here, I can see all of the colors up here, or all the colored residues are identified by their color. So you can actually see, if you're, yep. Yeah, hopefully. Let's see what we got. This is the notation you're going to start to see on post-translational modifications, and you can see the complexity in some of these sugars. These are really long-chain carbohydrates. Just make note of this as we get going. So basically, they're going to take all these sugars and they're going to put them out, and they stand out, and they're long-chained and they're branched on there. So, sure.
Okay, so now we're going to talk about a couple of the common characteristics about carbohydrates. So again, they have a very specific structure. They have 3 general elements: carbon, hydrogen, oxygen. They have, their main function is immediate energy, but they're often simple monomers or complex polymers. And here's a couple different examples, and that's really what we're going to go into now for the rest of the time. So there are a couple of different monosaccharide classifications. In general, you are looking at triose, which are going to be 3 carbon structures, and these are going to be your glyceraldehydes and your dihydroxyacetones. These are kind of your base level, as small as they get. This is what you're going to start with. And so you can see that ultimately you're going to have aldoses and ketoses. You can have your tetroses, your pentoses, but really the next big one that we're looking at is your hexoses. Just recognize that pentose is where your ribose and your xylose is going to sit, and your arabidose. So why do we care about ribose? Yeah, RNA, and then deoxyribose would derive from here too. So RNA and DNA kind of sit right here. But hexose is kind of where it's at. We have our glucose, our galactose, our mannose and aldoses, and our fructose and ketoses. Know the difference between aldoses and ketoses as being an aldehyde group versus a ketone group. Don't forget Fisher projections. Do you guys remember what Fisher projections are? Because inevitably they put on your test somewhere in this Fisher projection, and then you have to know what a Fisher projection is. So remember, Fisher projections are kind of these line-looking drawings and not the circularized ring form. So this is what you're talking about when you're talking about a Fisher projection. So here again is your aldose, ketose, triose, pentose, and hexose. And glucose is where we're going to spend most of our time here. This glyceraldehyde and dihydroxyacetone, these are going to be really important when we talk about glycolysis and some gluconeogenesis next week.
So you also know, yeah, go ahead.
For your final? So your final, you should just go back through the study guides from every single one of your tests, and then you'll have the final study guide, which will not be comprehensive, but will just cover the material that you have not yet been tested on. For a final? So that's your aldehyde.
Right. So, and then on the left one, on the ketose, that's ketone. So that's the important difference between aldose and ketose. So, and you will see that terminology, so you will need to know the difference between aldehyde and ketone group in that application.
Okay. So when we dig down into hexoses, really you've got your glucose, galactose, fructose, and then there's also mannose, which is kind of there but not really shown in a lot of the projections. So here's our glucose, and this is a comparison of them. Now it's interesting because fructose is the sweetest of all sugars. And what you'll see is that a lot of our foods are starting to have more high fructose corn syrup added to them. And this is what makes them, in some books and in some theories, particularly addictive, and particularly people want those types of foods because fructose is so much sweeter. And so you can see that here we have what actually happens with a fructose-rich diet, where you have a hyperinsulinemia, obesity, reduced energy expenditure because you don't have to use as much. But this is pretty interesting, and this actually, I don't know why the link didn't come up, but this is from a New England Journal of Medicine paper that tracked the increase in obesity with the increased use of high fructose corn syrup. So you can't really say that there's a causation, but you see this correlation. And so fructose is something to pay attention to. Galactose very rarely ever occurs by itself in nature. Usually it's going to be in a disaccharide, so it's complex with something. But when it binds with glucose, you get lactose, and that's really where you see galactose most of the time. And once it's absorbed, it's converted to galactose. Glucose is kind of our base level, and it gets complexed with everything for these disaccharides. So remember that in order to complex sugars together, you need a glycosidic bond. We won't go through that too much. Just remember that there's a glycosidic bond between carbon and a hydroxycarbon, and it's also an acetyl between the monomers. Also keep in mind that there's this hemiacetyl word that we're not really going to go into, but a hemiacetyl you'll see as an intermediate in some cases. So glucose plus fructose is sucrose, and so that's your table sugar. This is found naturally in plants, and it can be purified from plant sources. And then glucose plus galactose is lactose. Again, primary sugar in milk and milk products, but a lot of people have problems digesting a large amount of lactose, and we'll see why in a minute. And glucose is maltose. And that's shown here. And this is used in fermentation reactions. So here's sucrose. So you have your glycosidic bond. It's an alpha 1,2 glycosidic bond between glucose and fructose. Here's lactose. It's found at about 5 to 8% in human milk, 4 to 5% in cow's milk. And you've got, it shows all of your different bonds here, and you're using a beta 1,4 glycosidic bond for lactose. So this is the root of lactose intolerance. Lactose gets taken into the body. Lactase is the enzyme in our guts that will break it apart into glucose and galactose. And then glucose is used as a sugar. In lactose intolerant people, they're missing that enzyme, so they can't break it down, and then it's used as a source for bacterial fermentation, and that's what produces gas and all sorts of other digestive issues. So there's actually a pretty wide prevalence, and it's interesting to me because it's very regional. So this is a worldwide prevalence of lactose intolerance, and you can see that you have very high rates of lactose intolerance in this region of the world. So it's regional. And I don't quite understand that. And I didn't have time to look into it, but you know, summer reading. So here's maltose. Maltose is an alpha 1,4 glycosidic bond. So remember the different bonding patterns of these different disaccharides. And that takes us to polysaccharides. So polysaccharides, so you have your monomers, and now you have your di, and now you're going to have your polysaccharides. So again, you're using glycosidic bonds, and there's a difference. We have starches, which is a polymer of D-glucose. So here's unbranched and branched, just like we saw yesterday. And then you can have heteropolysaccharides, meaning that you have two different types, or you can have multiple monomer types. And so in starch, we see amylose and amylopectin, and we'll talk a lot about the difference between those two. So here's what a polysaccharide looks like. This is an alpha 1,6 glycosidic bond here, and then an alpha 1,4 glycosidic bond over here. So alpha 1,6 is at the branching point. Alpha 1,4 is the main chain.
Just to reiterate, how to put the glycosidic bonds, we will need to know how to name them, how they work. And they're also important. A glycosidic bond brand, the type of glycosidic bond is also important because not enzymes are very specific to the type of glycosidic bonds. So this causes disease states, right? So certain people can't debranch.
They can't cleave this.
They can't cleave it out to 1,6 glycosidic bonds, and they end up with glycogen deficiency. There's a whole class of glycogen deficiencies because they can't break that bond and process glycogen. So you need to know how to read that glycosidic bond. So an alpha means it goes down, and a beta means it goes up from that both structure.
That's a chair.
Chair, thank you. So that chair structure, alpha means it goes down often, beta means it goes up often. And you just count around from the oxygen in a clockwise fashion, that gives you your numbering. So from the oxygen, you go down from that ether, that's the first one, is the one that you go clockwise. 2 is the one with the, 2, and then the next one's 3, 4. That one, that one right there is the 4th position, right? 5th position is right there, and the 6th position is the one that's on.
It's kind of sticking off the top here.
Kind of sitting off that, off the top, not in the circle. That's how you determine whether it's an alpha 1,4 or a beta 1,4. That's how you count around sugar in order to tell where it's actually bonds ordered. And you don't care where it bonds on the second, on the higher end, right? So there might be a beta on there, but for all these situations, just for now, right? So it is more complex than this. For all things that we're working on, this one goes down. That's the important one. The alpha on the first one is
So the direction where you start from the first, if you go down, it's alpha, and if you go up, it's beta. So if this was 1 and this was 6, it would be beta. Make sense? Okay. So glycogen, as Ben was just talking about, there's a lot of different disorders of glycogen. But glycogen is a branched homopolysaccharide. So homopolysaccharide, how many different types of monomers does it have? It has 1. Yep. Homo means that it's completely homogeneous. It's got glucose. The glucose monomers are forming the alpha-1,4 linked chains. And then the branch points, and here's glycogen down here. So these branch points are an alpha-1,6. And they occur every 8 to 12 residues. And that's pretty precise. So every 8 to 12 residues, you're going to have a branch point. And then the molecular weights can reach several millions. These are enormous molecules. They're huge. And these are the main storage polysaccharide in animals. So here's an example of glycogen, and you're comparing it to amylopectin and amylose, which these two together form starch. So just keep in mind that the normal amount of glycogen in the body of a well-nourished adult is 350 grams, divided between the liver and the muscle primarily. So starch is a mixture of two homopolysaccharides of glucose. So here's starch, here's glycogen, which is really branched, and here's cellulose, which you can see is very, very ordered in pattern. So, and we'll talk about cellulose in a couple minutes. So again, amylose is the unbranched polymer, and that has alpha-1,4 linkages. Amylopectin is branched, and it uses, the branch points are an alpha-1,6, and it occurs every 24 to 30 residues, as opposed to the 8 to 12 residues. And amylopectin, again, very, very large because of all of its branching, up to 200 million, so it's a huge molecule. Starch is the main storage polysaccharide in plants. So glycogen in humans and mammals, polysaccharide is starch in plants. So this is just a quick look at the difference. Here's starch, which looks a lot more orderly, but you do have some of these branch points here. And then you've got glycogen, which is kind of disordered. So starch is amylose and amylopectin. You can see those up here. We're going to see all of this here in a few minutes, but yeah.
So the thought exercise, I think it's really important to note that they're all the same thing. All of the differences are where they branch and
What the different residues look like.
yeah. And so one, I need to make one other point because I don't think it makes sense. The number 1 carbon is a chiral center. You'll want to know that. That's called the anomeric carbon.
You done? Okay, so it's an anomeric carbon. And that's carbon number 1. So we have amylose and amylopectin. They have a lot of these non-reducing ends and then a few reducing ends, and you have the alpha-1,6 branch point here. So these non-reducing ends and reducing ends lead to a lot of kind of structural organization. and it changes the way we metabolize things. So as Ben was just pointing out, these are all made of the same thing, and yet we can metabolize some and not all. So glycogen and starch both form granules a lot in cells. And these granules have enzymes that can synthesize or degrade the different components. And because the glycogen and amylopectin only have one reducing end but a lot of non-reducing ends, that changes the way they're going to get degraded and synthesized. And then remember that here's cellulose, which we saw before, is this highly ordered structure. And what kind of bonds are here? Hydrogen bonds. Hydrogen bonds are going to change the way things pack together. So as we saw before, we have these branch points. Those are covalent. Those are glycosidic bonds. And then you have hydrogen bonds that are holding things together. So humans and a lot of other animals can't digest cellulose because we can't cleave the beta-glycosidic bonds. And this is why. It's all so patterned and so structured. So here's kind of your cheat sheet, the difference between cellulose, starch, and glycogen. It tells you what kind of subunits they're using, what kind of bonds they're forming, how many branch points they have. Here's a diagram of them, and this is what the shape looks like. So just kind of your cheat sheet for you. Any questions? Okay, let's take a quick break, and then we'll come back and look at glycoproteins.
So I got that.
What? Okay, so now we're going to talk about glycoproteins. Now, why not?
To annoy you?
Why
So
not?
if you see something on the exam, you will see something less expressive than non-reducing ends. Almost always. They love that. So non-reducing ends. So that's two non-reducing ends. Sucrose is a disaccharide.
What's it a disaccharide made out of? Glucose, and what's the other thing? Nope, that's lactose. Remember, it's got the lactose in the end. It's fructose. Yep, glucose and fructose make
They love
sucrose.
sucrose and the two non-reducing ends.
Yeah, we didn't mention the two non-reducing ends versus just one. So starch, remember, it has just that one non-reducing end and the one reducing. So, okay, so glycoproteins is just a protein component that's got basically carbohydrates attached to it, and that gives you a glycoprotein. Remember, the carbohydrate is attached via the anomeric carbon, has been mentioned, and about half of all mammalian proteins are going to be classified as glycoproteins, which is really, really important when we talk about it from a biotech standpoint, because a lot of times we are trying to make these proteins, right? We're trying to make them for drugs, we're trying to make them for a bunch of different things. Well, the cheapest and easiest way is to use bacteria to make it. But guess what? Bacteria don't add glycosylation residues. They can't add sugar residues to proteins. So you can't make, if you have a protein that has to have glycosylation in order to function, you can't use a bacterial system to make that. So you can see that we've got different systems shown up here. Humans have the most complex glycosylation patterns. Here's the names of a bunch of the different sugars over here. So here's your key. And so humans have this very complicated strategy of glycosylation patterns. And this fucose, not to be confused with fructose, fucose, if you are fucosylated, this is a huge key for immune recognition. So fucose and then sialic acid, which you can see on the ends of all of these different branching chains here. And you very rarely see them on the ends of other branching chains in other species. So sialic acid and fucose are major components to determine self and non-self. Insect cells can add fucose, which is weird because animals don't, and plants can in some cases, but plants can't add sialic acid, and insects can't add sialic acid. In fact, insects use what's called a high mannose structure, which means it's almost all mannose at the end. And so that doesn't allow you to do everything you need to do. So just keep in mind that bacteria really don't glycosylate. There's some evidence that certain species can glycosylate a very few number of proteins, but in general, they don't glycosylate. And that the viral proteins are heavily glycosylated, and that helps them evade the immune system, and we saw that with HIV. And then just think about, here's different patterns that come from different species, and different species can't use all the same sugars. So xylazine over here, you can see plants use that, and they are unique. Nothing else uses that. So let's talk for a minute about erythropoietin. So have you guys heard of blood doping in the Olympics and cycling? Cycling's a big thing for blood doping. Ben, shh. So here's a quick story about it. Lance Armstrong, you guys know Lance Armstrong, right? He was stripped of his 7 Tour de France championships because it was shown that he used EPO. Now, EPO is going to go to your bone marrow space, and it promotes the creation of new red blood cells. If you have more red blood cells, what can happen? You carry more oxygen. So you have an increased amount of oxygen. So you'll see blood doping was also considered not just EPO usage, but first it started with athletes would have transfusions right before their event. So then they thought that that wasn't a very good idea. So then they started using EPO. And they came up with a synthetic version of EPO, and that's kind of the cool part, because EPO is something that you naturally produce. So how do you know if somebody is using EPO that is not theirs? How can you distinguish, right? Because you're going to test the blood, and you're going to say, what's the level of EPO? So how do you know if it's EPO that you've produced versus EPO that you've taken? And the answer is glycosylation. So here is the natural EPO molecule. And you can see all these different sugar chains here. We have our sialic acids hanging out over here. We have our GalNAcs. We have our fructose. So this is the synthetic version. Now, of course, they didn't make things the same color, so it makes comparison a little bit harder. But you can see that we have some slight differences in the way that this is structured and the way that the sugar residues are coming off. So you'll see this one difference here. You've got 2 galactose, and then you've got sialic acid on both ends. Here you have a galactose and a GalNAc, and then 2 sialic acids. So you have very subtle differences between a synthetic EPO and your natural EPO. And so in order to determine if these athletes were actually using EPO and doping blood, they detected a glycosylation pattern, which I think is really cool, that they could detect something with such specificity that they can find just small differences in the way that these are made. So here's the natural, and you can see that we have a lot more glycosylation on the synthetic. Oh, good question. So negative side effects of doping. Because you're increasing your red blood cells, your blood gets thicker. That's one of the big consequences, which makes your heart work really hard to pump thicker blood. So that can be one negative consequence. There's also been some evidence that it has been linked to cancer-causing agents. Because everything is, you know, so why not? This too.
Honestly, I don't know that I necessarily have too much trouble with EPO. If I was sitting around, if you're asking me, do I care whether my entire son uses EPO when he's doing it for fun? I don't really care.
because everybody does.
It's natural. It's not like you're, yeah, well, not just that, everyone, I mean,
So there's like a huge history of this throughout the Olympics, right? And in one year, they like disqualified the whole Russian team because, was it? Well, there you go.
if you want an interesting documentary on this, look up Icarus on Netflix. You'll see a very interesting story on this very topic. Sure.
Yeah, naturally someone could. So here's the other question. Is EPO affected by training at higher altitudes, right? What if we train, Ben and I used to live at 7,200 feet. Laramie is at 7,200 feet. This is not. This is, Fargo is not. I'm just telling you that right now. So we used to live in the mountains. So did our EPO, did we produce a different amount of EPO? Did our red blood cells carry oxygen differently? Yes, probably all of that happened. There's some pretty interesting studies on this. And EPO, because it's got such a cool story, if you're still looking for something to do a video on, EPO might be a cool one to look at. Because you can tell a really cool story with the glycosylation sites.
So do athletes think they don't get tired?
Yes. Yeah, because they can carry more oxygen, so they increase the number of red blood cells, so that they can carry more oxygen, so their muscles don't get as fatigued.
Does that beat rate of their heart beat harder?
Yep.
With blood clots,
Mm-hmm.
We'll discuss that topic next week. We'll talk about lactic acid. We'll talk about lactic acid 2 weeks ago, right?
We mentioned it briefly, yeah.
So we'll actually talk about why this is really important, because then you don't get lactic acid buildup, your muscles don't hurt, all these kind of things are really critical for this.
So you also have to keep in mind that when they're blood doping, they're not doing it chronically usually. So usually they're doing it right before a race. So you strengthen your heart, so it's nice and strong, and then you dope right before. So they train under like a little bit hypoxic conditions, so it's almost like training at higher altitude because it strengthens your heart. And so then when you drop down in altitude, in theory, it should be very efficient. And so then they dope right before the race to increase their red blood cells. Now, we can have conversations about how quickly that effect happens. I honestly didn't look into it far enough for me to tell you how quick that effect happens and how long it lasts. So I don't know if, the Tour de France is a very long race. I don't know if he had to do multiple injections or if it was like one at the beginning and then, so yeah, I haven't looked into the story that much. Really, my interest comes from just look at the difference in glycosylation patterns. And we can detect this based on sugar residues, which I just think is fascinating. I think that's really cool. And it goes to show us how much these glycosylation patterns change immune recognition, right? So antibodies recognize these specific glycosylation patterns as being self and non-self. I don't know.
That'd be a really interesting question to answer. Whether this would be a mass spec-based assay for the two, or whether it be an ELISA or Western normative type differences.
It's a really fascinating story. I wish I had had a little more time to go into it. But alas, I did not.
No, I think we could 100%.
All right. So there are a couple different kinds of glycosylation you have to be aware of. There's O-linked glycosylation and there's N-linked glycosylation. And these fall under this huge umbrella of post-translational modifications. So your O-linked is going to link through serine or threonine, and it links through your hydroxyl group here. So here's your O-linked glycosylation, and then your N-linked is going to link through asparagine, and it's going to link through that nitrogen. So again, here is your first post-translational modification, O-linked glycosylation, serine, threonine, tyrosine.
And you notice the previous slide just said serine and
Threonine.
right? Those are two traditional O-linked. You'll see in the places have different, as the research is expanding,
So remember, you're linking through an oxygen. That's why it's called O-linked. And so these are going to help in protein folding, protein localization. They can be structural in some cases. And you can get all these different sugar residues building off of that. Then you have end-linked glycosylation. So again, you have to have that charged N group or that charged nitrogen. So you're looking at your lysine and asparagine. Again, protein structure, protein folding, protein localization. And what do we know about protein structure and function? They're related, right? And so if we can't form the proper structure, we don't get the proper function. And so that's why it becomes very important, going back to the previous slide that I showed you, about all these different species are able to do with glycosylation events. Because if we try to make a protein that requires a sialic acid and we try to do it in a plant, well, that may or may not work. And in bacteria, it definitely won't. So these are common post-translational modifications. You need to be aware of all of these. So we've kind of talked about a lot of these. We have our phosphorylation, and underneath you'll see the residues that these occur on primarily. So phosphorylation, methylation, acetylation, and hydroxylation, and you can see what that post-translational modification is. Now, post-translational modifications help with the folding of the protein, so the structure and function, but they also can sometimes stabilize that protein. So it stays around a little bit longer, not like our progesterone that has such a short half-life that it goes away right away.
I posted this chart on Blackboard.
So you now have access to this chart as
You
well.
should know this chart because it'll be stuff exactly on the test.
So then the formulation becomes really important because remember that FMET that we talked about when we talked about translation and how we have to start with a specific methionine to get our right reading frame. Well, this is where that FMET comes from. So that's a formulation event. That's a post-translational modification. So you have palmylation, pronation, myrcillation, ubiquitination. This is the key that says come and degrade this protein. So this is kind of that terminal degradation signal if you add ubiquitin. Why not?
So pollination, prenylation, risk of elimination, or us, risk of whatever, you know what I mean?
Yeah, the M1.
Those are all used to anchor proteins and membranes.
So we're going to see those
One of
tomorrow
them,
more.
We'll see, yes, we'll see more mechanisms, but those primarily are also used to anchor loosely a protein in a membrane. The other ones are all used for structural modifications, so you use something like phosphorylation. Phosphorylation is done by a kinase. That can change the function, that can activate an enzyme, that can do any number of things. Methylation, you're going to go into this, I'm giving out myself, right?
Actually, no, I was not. Go right ahead.
Okay. So methylation can be used like histones to activate and deactivate DNA. You might recall DNA has same acetylation. Those can be used to activate and deactivate.
Remember, those are kind of our on-off switches, is this top row here.
So
These are our on-off switches.
ubiquitination, I don't think I talked about. I think we're going to talk about.
No,
So I think I talked about it briefly when I talked about parkin. So ubiquitination is when we add ubiquitin to a protein, and they usually add in specific numbers, usually a lot of them, and that will signal a protein for degradation. So parkin comes along, notices a misfolded protein, and it gets ubiquitinated, and then that signals that protein needs to be degraded and moved to the cell. If you have a misfold or a genetic mutation in parkin, then ubiquitination cannot occur efficiently, and that's when you start getting protein aggregation. That's what Parkinson's, one of the ways that Parkinson's goes into.
okay.
Simulation is also related to ubiquitination.
Yeah, these two are related. So these are the crap, I screwed up and misfolded a protein and I need to get rid of it. That's these. These are the on-off switches. These are the let me anchor something. Formulation is kind of an on-off switch. It's kind of a beast all of its own. And then these are the self-recognition. So these are the carbohydrate ones. And you can see down here, I'll just point out that these are protein groups. These are functional groups up here. And then these are lipids. And we know that lipids are going to help anchor things in membranes. So that might help you realize and remember which ones are used for anchoring versus which ones are functional groups, are usually the on-off switches. And then your carbohydrate groups are recognition, and these protein groups are usually degradation. All right, so proteoglycan, again, this is a tetrasaccharide bridge. That's the main thing that I want you to be aware of here is this tetrasaccharide bridge. And then here's your core protein here, and this tetrasaccharide bridge is linking off of serine, which tells you it likely what kind of a link. Is it O-linked or N-linked? You think it would be N-linked? It would have to be O because it's coming off oxygen. So remember O for oxygen, and serine has that OH that's sticking up on it. So yep, so that one's going to be an O-linked most likely. And remember you have a beta-1,3 and a beta-1,4 in between the galactose and your xylose, and a beta-1,3 here, beta-1,4 here, and here's your chondroitin sulfate. And we know that chondroitin sulfate is one of those molecules that helps lubricate our joints. Yeah, so again, here's another one that we know quite a bit about. This is a different way of looking at this proteoglycan, and sorry, this didn't show up very well on the screen. But aerocans we know a lot about. It's probably what we know the most about, and it forms these brush-like structures. And then it's attached to hyaluronic acid. And you guys may have heard of hyaluronic acid before because we use it for all sorts of things. Somebody might actually say, I'm going to take hyaluronic acid to try to improve their joint mobility.
It's a huge percentage of the human body is hyaluronic acid. It's that hydrogel, and you should already discuss hydrogels. That's what we're good at.
Hydrogels? Yeah.
So a lot of carbohydrates, a lot of hydrogels will get at carbohydrates.
They form these cross-linking networks, and it's basically a gel that stays in place. And we'll talk a lot about it when we talk about drug delivery, so I didn't really cover it in this class.
Carbohydrates have the hydroxyl group on them, so they're very easy to make them bond with water more readily, so they keep things moist.
And so they'll swell. Water will pull into it.
Things hydrated, that's why you see a lot of that's why you see a lot of hyaluronic acid, water around, and obviously the cells like water, and also one of the things that we use tons of other applications for hydrogels that are based on carbohydrates.
Yeah, so a lot of your drugs will love being in those hydrogels, and then as the hydrogel essentially dehydrates, the drug comes out, or as it swells, it forces the drug out. And so there's some pretty interesting drug delivery pieces with hydrogels. I think that's everything we wanted to say on that one for now. So just remember here that you have this anomeric carbon of xylose is linked to the serine hydroxyl group. So because this is a serine hydroxyl group, this is likely an O-linked glycosylation event. So we also can sulfate these glycosaminoglycans. So we've talked a lot about, and you guys have probably heard in, just out in popular culture about glycosaminoglycans, because we think that we can take glycosamine and chondroitin and actually improve some of our joint functions. So here we have sulfated glycosaminoglycans, and they have this large rod-shaped protein. So syndicans is one of those. It has a single transmembrane domain, and glypicans is the other one, and it's anchored in the lipid membrane. So if we look at these couple of different ones, syndican actually will grab heparin sulfate, and we'll talk about heparin in just a minute, And it will also grab chondroitin sulfate, which we've already talked about. And then you have this core protein. And so in Syndecan, your core protein goes through the membrane, so it's a transmembrane, as opposed in Glypican, where you've got kind of this globular domain, and you have 14 serine residues, which give you these disulfide bonds here. And then you have a GPI anchor into the membrane. So you're not actually going through the membrane here, but it's covalently attached to a membrane lipid. And in the Syndecan, it's held in place in the membrane because of hydrophobic interactions. So hydrophobic interactions are going to keep Syndecan in place, and then it's going to get decorated by heparin in some cases, and heparin is going to be used to prevent blood clots. So here's heparin. It's a repeating polysaccharide that gets sulfated, and because it gets sulfated, it has all these negative charges. So heparin sticks to a lot of different things. Heparin sticks to a lot of different growth factors. So we've talked a little bit about growth factors and cytokines. Remember, your cells are putting out these growth factors and cytokines to say, hey, I'm here, or go do this. And so it begins a lot of signal cell transductions. And what we see is that there is a small little amino acid motif in these growth factors and in a lot of molecules that bind heparin, and they're highly positively charged. And so they're made a lot of lysine and arginine, which both have the positive charges, and then they react with the sulfate groups on heparin sulfate. And because these have the negative charges, you see electrostatic interactions for heparin. And that's how heparin is binding, is through these electrostatic interactions. And then it disrupts the coagulation cascade. So this is what that looks like in terms of if we draw it out in this particular type of notation, where you have your sulfate groups here, and you can see how many sulfate groups you have.
You should know a little about heparin. What are your exercises?
So hyaluronan, we talked about hyaluronic acid. Hyaluronan is the protein. So you have hyaluronan and aggrecan, which we talked about as being this brush structure. They form these non-covalent aggregates. And when you mix a lot of collagen in here, you can create those hydrogel type networks. And so you can see this is a network that's depicted here, and you've got a linker protein, you've got a lot of aggrecan, and it's swelling, so it's drawing water into that. And so again, your cells like to be in there, and we can use that property of drawing water in to deliver different drugs. It also covers the joint surface of articular cartilage, which allows it to reduce friction and balance your load. So it's this very low friction molecule because it holds so much water. So it provides lubricant. Now this is, I didn't talk about it, but basically this is very similar to the way mucus works. So if you think about your lungs, if you think about your mucous membranes, this is very similar to the way mucus works. Think about it as working like a hydrogel. It's pulling water in, and it's lubricating. So here's just another look at it. This is keratin sulfate here with your link proteins, chondroitin sulfate, and together this is the aggrecan core protein, and then you have hyaluronan here as the backbone, and that's your disaccharide. And you can actually see this under a microscope, which I just think is so cool. I really would kind of like to look at these at some point. So they look like these little brush structures, and those brush structures are going to hold stuff right to the surface of this molecule. So when we think about it, here's a joint with osteoarthritis. So there's a large percentage of people that are suffering with osteoarthritis in the United States, and in fact worldwide, but it's a very large population. What you end up seeing is the cartilage wears away at the ends of the long bones, and here we're just looking at just a specific joint. You have the synovial membrane and synovial fluid, but you have little pieces of bone, you have cartilage, and so there's been some evidence that taking glucosamine and chondroitin will actually help improve the pain from osteoarthritis. And in fact, we give glucosamine and chondroitin to dogs sometimes if they have joint problems. And for large dogs, you can see that some of the dog food is actually supplemented with glucosamine and chondroitin. So there's this large body of anecdotal evidence that suggests that it will help with osteoarthritis pain. But again, New England Journal of Medicine, that shows us that here's your placebo, here's glucosamine, here's chondroitin, here's the glucosamine chondroitin, and here is an anti-inflammatory. So they're looking at it after 4 weeks and after 24 weeks, and their conclusion was that there really is not a tremendous benefit in terms of reported pain. So they're talking about patients self-reporting pain. So there's not a tremendous difference for each individual one above the placebo, and that there may be a very small difference if you take them together. So why would this matter to you guys? Future pharmacists, why would this matter? Yep. It happens a lot, yeah, because people are looking for more natural type remedies for pain. And so this may work in combination, but if they take glucosamine by itself or chondroitin by itself, the evidence suggests that they're not going to see an appreciable benefit out of each of the individual components. But if you take them together, you may have a slight benefit. So the other big piece of glycosamine and glycans, man, there's this, he left and now I can't talk, I don't know, is chitosan. So chitosan is a really interesting molecule that's approved for certain medical uses, and it comes from the shells of arthropods. So you have chitin, and then it gets deacetylated and goes to chitosan. And this is being used in a lot of different biomedical applications for its ability to deliver drugs. It has its own natural antimicrobial abilities, and so chitosan is definitely becoming something that's it got a lot of potential for biomedical applications, but it's very hard to get it pure enough to use for medical applications, and to understand how it's going to break down, because how it breaks down depends on its deacetylation. So it gets acetylated and deacetylated, and its degree of deacetylation tells you how quickly it's going to break down. So that brings us to glycolipids, which we touched on yesterday a little bit. Again, glycolipids are a lipid backbone as opposed to the protein backbone that has a glycan or an oligosaccharide that can be covalently bound. So again, these are going to be your structural pieces. So they're going to be parts of membranes. These are going to be in your blood groups. And in gram-negative bacteria, it covers lipopolysaccharide. Now, lipopolysaccharide is the molecule that your immune system is actually responding to. When you have an immune response to a bacterial invasion, you're responding oftentimes to lipopolysaccharide. So here is lipopolysaccharide. Here's the O antigen form. Here you have a core polysaccharide, and then you have this disaccharide diphosphate, and then you have fatty acids that are coming off. So this is the structure of lipopolysaccharide, and this whole piece is called lipid A. So we think that lipid A is probably what we're responding to, and oftentimes. But the interesting thing about lipopolysaccharide, and this is just a reminder of the difference between gram-positive and gram-negative bacteria. So we think that oftentimes this lipopolysaccharide is almost impossible to get rid of. So to get it off the surfaces is very, very difficult. You have to use like an acid or a base wash to get it off of glass in the lab. It's everywhere. And in fact, we did a pretty interesting study where we looked at nanoparticles. There's lots of uses of nanoparticles. You guys are exposed to nanoparticles all the time, and you probably don't even realize it. But one of the questions we had is, nanoparticles, because of their surface area, are thought to be really good drug carriers. But if they can carry drugs, they can also carry toxins. And if you don't process them and synthesize them in a completely clean environment, they could actually be contaminated with LPS, or lipid polysaccharide. So can they deliver a toxin? And so that's what we looked at. And we tried to figure out if they could, in fact, deliver a toxin, because we know that different things can absorb on them. Turns out it's a very difficult question to ask, and we can go into that another time. But just know that lipopolysaccharide absorbs to all sorts of things. So in bacteria, lipopolysaccharide is part of this kind of what we kind of consider our extracellular matrix. But you guys all have an extracellular matrix that surrounds all of your cells and really dictates what kind of interactions we see with the drugs that we use, with the biomedical devices that we put in, and the way cells interact with each other. So again, this is your material on the outside of the cell. It provides strength, elasticity, and a physical barrier. It's mostly made of proteoglycan aggregates, collagen, and elastin. And again, it can be a barrier. It can be very, very important in preventing metastasis of tumors. This is what it's going to look like here. You have syndicans. And remember, syndicans are going to have one transmembrane domain and go through. You have these integrins, and integrins are going to interact with proteins like fibronectin and actually help mediate those cell signaling cascades. So integrins we're going to talk about a lot tomorrow. And they're receptors for proteoglycans. Again, they're going to link this extracellular cytoskeleton, which is what we can think of our ECM as, to regulate growth, mobility, cell death, and wound healing primarily. So here's your extracellular matrix. And then we also have oligosaccharides and recognition, which we've been talking about kind of all day today, how important these are in recognition. We saw it with GP120 that connects with GP41 for HIV. And then you interact with some of those cell surface receptors on the CD4. So here's our virus that shows us our interaction here with our oligosaccharide chain that sits right in here. Here's our glycolipid here. We have a leukocyte, which is an immune cell. Here's our bacteria that has a receptor that now interacts with our cell. So we can have cell interactions here. We can have cell-bacteria interactions, virus-bacteria interactions. So you would think that all of these interactions would actually prevent bacteria and viruses from getting in. Well, we've seen with HIV how viruses kind of get around that, right? They mask that with glucose residues and with different glycosylation events. And then they leave the one recognition site open. So we saw that, but what do bacteria do? Bacteria have adhesins. Adhesin proteins are very similar to our integrins that we just looked at, where integrins actually are mediating that interaction. So these are adhesin proteins that are on the surface of the microbe. And then here's our toxin, which is going to be like our LPS. Now, our microbe can actually sit down, and it puts out these pili, right? So these are kind of those little tails that come out, help bacteria be motile, help them move around. And those have these adhesin proteins, and in this case, they're called lectins. And those interact with the cell membrane. Again, those adhesins bind to a lot of different copies present on the host cell and allow the microbe to overcome that host cell defense by binding to so many different copies. In fact, we can think about that as like a competitive pathogen binding. So here's our microbe right here, and we can see that they have essentially 4 different ways that this can be considered competitive. And each one of these, here you have a microbe that has a similar kind of ligand that would bond onto your adhesin or your adhesion receptors here, and it could take the place of the normal host molecule. In this case, again, you have a slightly different kind of bonding that takes the place. Here, we can actually sit down inside here in multiple cases, and in this case you have an antibody. So again, we're going to compete for that binding site. Now there are lots of different ways that we can look at these glycoconjugates, and we've done a lot of different things to try to help use glycoconjugates to prevent this microbial binding and prevent microbes from being able to enter cells. Here are just a couple of different ones. Essentially, you can see that we have a lot of kind of a globular protein, and then we have some reaction sites around the side. Here's a glycodendromer, where you've got your dendromer is this highly branched thing here, and then you've got glycosylation all on the outside. You've got your glycopolymers. Here's our nanoemulsion. Emulsion indicates it's going to be like a lipid almost thing, where we have our hydrophobic tails and our hydrophilic heads on the outside. Here we've got a glyconanoparticle, so we take a nanoparticle, in this case it's probably a gold nanoparticle, and we put sugar residues on the outside. And remember, we can now, if we use these different molecules, we can think about how we can compete and we can protect these adhesion sites by binding to the microbes and binding to those microbes, to their adhesion proteins and to their lectins and their toxins. And then if you guys want to look at how Ben did his pymol activity, I put all of that up here so you guys can try that, because he was doing things that were a little bit more advanced today. So here's all of that information up here. Do you guys have questions? Yes. A micelle? So a nanoemulsion and a micelle is a type of nanoemulsion in a lot of cases. So in some cases, you will see a micelle will have, you can have this hydrophobic kind of shell layer and then hydrophilic, so you can encapsulate two different kinds of drugs. In this nanoemulsion, as it's shown here, you could only encapsulate hydrophobic inside the center, but they're very, very similar. Good question. Yeah, so let's go back to that. Okay, so remember how Ben showed you that we've got GP120. Actually, let's start here. So we have our envelope, right? So the envelope is going to encapsulate the virus. And on the outside, you have GP120. And so your GP120 is going to form a complex. And then it complexes with GP41. So remember that very first gel where I showed you we were seeing a conformation in Western, and I said, make sure you see this GP120 and the GP41. That's because those are these proteins here. So these are the envelope proteins that the HIV virus uses. So GP120 complexes with GP41, and then they stick out of this viral envelope on top of the, essentially on top of the virion. So that's this part here. So then 120 interacts with 41, and then that creates this mesh. And then they add glycosylation residues on the outside of that mesh because that will hide the, it basically hides the virion or hides the virus. But the one place that they don't glycosylate is right here. And it's hard to see in this mesh, but they leave this part open because this part interacts with the CD4 receptor. So the CD4 receptor is on your T cells. So that's our marker that it's a T cell. So essentially, your GP120 and GP41 can go down and sit on CD4. And when they sit down on CD4, they can then fuse with your cell, your T cell, and go into the T cell. Does that make a little more sense? So it's in this example right here, it's this yellow part that is basically unblocked. And it's the only thing it can interact with is CD4. Okay? Any other questions? No? All right, well, we'll end a little early tonight then.